Hi there, and welcome to lesson 5 of the Unit 1 of the National 5 biology course. Lesson 5 is all about genetic engineering. Okay, so it's a really interesting topic, but it's quite a short one in terms of the amount of content in it. So we'll go through all of that. We'll check that you understand all of that, and then we'll move on to looking at how it's applied to exam questions later on as well. So genetic engineering is the ability to transfer genes from one organism to another. Okay, so as it says here, genetic information can be transferred from one cell to another using this process of genetic engineering. Okay, so transferring a gene from one organism or one cell to another. Now, this process involves the use of enzymes. And the reason it involves the use of enzymes is because we have to both extract and insert genes. So we're doing a degradation, a breakdown reaction, and we're also doing a synthesis or a build-up reaction here. Okay, so we're extracting and inserting the genes here. Now, the enzymes carry out that by cutting here and here on the source chromosome, okay, the source bit of genetic material, and extracting this gene here from the rest of the material, okay, because that's the gene of interest. That's the one that we want to transfer. And then it takes that gene over to the other organism. Now, in this example, the other organism is a bacterial cell. Now, as we know, bacterial cells do not have a nucleus. Instead, they have a circular plasmid, which contains genetic material. So what we're able to do is we're able to cut open the plasmid, okay, and cut it open, and we can then insert that gene into the plasmid. And what we're left with is this thing here, which we call a modified plasmid. It's been modified genetically. So we've added the gene of interest into the bacterial cell. Okay, so we've taken that gene there from whichever organism it happens to be, and we've extracted it using enzymes, and then we've inserted it using enzymes. Okay, so that's the two key. I mean, it's a lot more complex than this, the process, but that's the key take homes. Okay, enzymes are used in the process to both extract and insert genes. Okay, so if we move on to the next slide, and we're going to look at the specific stages of genetic engineering. This is probably what you're asked the most about in exam paper questions on this topic. And having a good knowledge, the ability to memorize and to recite the steps involved in genetic engineering is going to be a huge advantage to you if you get a question like this in the exam. Now, although it looks a little bit daunting with all that text on there to learn all of this, it's actually not too bad in terms of learning it once you get going and once you become familiar with the terminology, with the words that are involved. So the first thing that happens, and I mentioned it a moment ago, but we need to identify a section of DNA, okay? We identify a section of DNA, and that section of DNA will contain the required gene, okay, from something that's known as the source chromosome, okay? So, for example, here it says the chromosome is extracted from the human cell and the required gene located. So that's equivalent to this, okay? That section there is what it's talking about here. We then need to extract that required gene. Now, we spoke a moment ago about the fact that enzymes are responsible for extracting and inserting genes. So it's an enzyme which is responsible for extracting that required gene from the source chromosome. Okay, and we can see that here in step 2. Okay. Then we need to place that gene into another organism. And as I said earlier on, if we're using a bacterial cell, the place where we put that gene into is the plasmid of the bacterial cell. Okay. So we need to get a bacterial cell that carries the plasmid, and we identify that. Then we extract the plasmid from the bacterial cell. And then that allows us to then insert the required gene into the bacterial plasmid, okay? So we can insert the required gene into the bacterial plasmid. And again, insert, extract, we use enzymes for those processes. And we use the same enzyme because, I mean, again, without going into the technical details of it, it means that it's more likely to be able to bind to the new chromosome that has been attached, or the new plasmid it's been attached to. Okay, so extract the required gene using enzymes, insert the required gene into the bacterial plasmid. Okay, so that's step 3 here in our staged process. Okay, and I should pop this arrow here just so you know where that one goes as well. Okay, so hopefully that this isn't seeming too daunting so far. We're establishing all the keywords and we're putting them in order, and I think logically it makes sense. I'm going to show you a diagram in a minute to help you as well to visualize this. But the last step here is to insert the plasmid. So this is the plasmid that we've put the required gene into. We put that plasmid back into the host bacterial cell. Okay, so we put it into the host bacterial cell, and that allows us to produce a genetically modified or a GM organism. Okay, so we insert the plasmid into the host bacterial cell to produce a genetically modified organism. Then what happens is they will generally just leave the modified bacterial cell to multiply. And as it multiplies, each of the new cells will have that required gene and they will produce the protein that is correspondent to that gene. Okay, and it will produce it in large quantities as well. The classic example of this is human insulin that type 1 diabetics like myself inject every day. So we're doing 5 injections a day, so we need a lot of insulin. We go through it quite quickly. And the way in which they produce that insulin is through putting the human insulin gene into bacterial cells and allowing those bacterial cells to produce human insulin in a huge fermenter. And they collect that and distill the insulin away from everything else, and they bottle it up, and that's what us diabetics inject into ourselves to keep us well. Okay, and we zoom in here. We can see a nice wee diagram which shows us those stages again. So stage 1 here, we've got our bacterial cell that contains the plasmid. We've got the human gene which has been extracted from the source chromosome. Okay, if you imagine the source chromosome over here. Okay, the source chromosome is probably a little squiggly thing over there. And we zoom back in there. Okay, so we've got the human gene from that source chromosome. That gene then needs to be inserted into the plasmid. So we need to open or cut open that plasmid so that it can accept that gene. We then insert the desired gene, the required gene, into the plasmid using enzymes, so that now we have what's called a modified plasmid here at stage 3. It's been modified. It's had that required gene added to it. The next step is putting that modified plasmid into the bacterial host cell, and we pop it in here like so, like we can see in stage 4. And then that genetically modified organism, that bacterial cell, will be able to produce the protein that the gene codes for. Okay, as I say, that can be human insulin, it can be a human growth hormone, it can be other things as well. Lots of other things, quite often hormone based. Okay, so that's the process, and you can see here the bacteria multiplying in these final stages to produce large numbers, so that it can produce lots of our product, our protein product from the gene. Okay, so hopefully that all makes sense in terms of the stages involved in this process. Feel free to pause this to write this down, to copy it down, to look through your notes, whatever you need to do in order to remember this process. But if you can remember these steps here. Okay, and the keywords, you can see I've put them in bold here. If you can remember those keywords as you're describing it and use those words, really after a while, you'll start getting used to it, and you'll actually find this relatively straightforward, I hope. They can ask you to write maybe a slightly longer answer on this topic, just because of the nature of it. It's a step-by-step process, so it lends itself well to maybe a longer question worth a few marks. So it's well worth, I think, memorising this and making sure you can recall it. Okay, so we're finishing off the lesson part of this topic, and we're going to move now on to the past paper questions. Okay, folks, so past paper questions for the genetic engineering topic. Let's have a look at some of them. So this one here, it says the diagram below represents part of the process of genetic engineering, and we can see a diagram very similar to the one we shared in the lesson there. It says A part 1, structure X is removed from the bacterium and modified during this process. Name structure X. So if we look up here, structure X is from the bacterial cell. It's circular. So the thing that we've learned all the way back in topic 1.1, and we learned a bit more about in this topic 2, is the plasmid. Okay, structure X is the plasmid. Part 2, the bacteria have an initial concentration of 1,000 cells per centimetre cubed. Each cell divides once every 30 minutes. And calculate how long it will take for the concentration to become greater than 15,000 cells. Okay, so for this question, it's a case of doubling. Okay, it's a case of doubling. So if our first number is 1,000, we'll jot down 1,000 there. After one division, we will have 2,000 cells. Okay, and after another division, we will have 4,000 cells. Okay, after another division, we will have 8,000 cells. And after another division, we will have 16,000 cells. Okay. Now our target was to get to 15,000 cells. And it tells us that the cell divides once every 30 minutes. So what we can see here is we've got 1, 2, 3, 4 divisions were required to get to 16,000 cells or above 15,000, which was our target. So we needed 4 divisions to do that. Now it tells us that each cell divides once every 30 minutes. So that means for every division, we have 30 minutes. Okay. So that'll be 30 minutes for the first division, 60 minutes for the second, 90 minutes for the third, and then 120 minutes for that fourth division there. Now, what is 120 minutes? Because it wants the answer in hours here, you'll note. Okay, what is 120 minutes in hours? Well, there's 60 minutes in an hour, so that's going to be 2 hours. Okay, so that's how we work out that question there if you happen to get it in the exam. Question 2 is very similar to the diagram that we shared again in the PowerPoint. We have to complete the boxes to describe each of the steps carried out to transfer a human gene to a bacterial cell. So for this one, we need to pop in our answers into the relevant boxes there. So I'm just going to pop a text box over the top here so that we can type in our answer. Okay, and for this one, for step 2, we have already extracted the chromosome from the human cell and we've located the required gene. The next thing we have to do is extract the required gene. Okay, we have to extract the required gene. Okay, so that's that step there. This time we've identified the bacterial cell, that carries the plasmid. And if I pop another wee text box in here, there we go. We need to extract the plasmid from this cell. Okay, so we extract the plasmid from the bacterial cell. The next step that we need is going to be in step 3 here. So we've now got the required gene. We've now got the plasmid. We are going to insert the required gene into the plasmid. Okay, so that's step 3 there. And then step 4 is going to be, we're going to insert the modified plasmid, okay, the genetically modified plasmid into the host bacterial cell. Okay? So we're going to insert that modified plasmid into the host bacterial cell. So an answer something like that would get you the marks for that question there, folks. Okay, so have a wee look at that. Check it against your notes, see where we've got the answers from, and hopefully that'll start to sort of be making sense, this whole process. Okay, 2B, name a human hormone that has been produced by genetically engineering bacteria. Okay, I mentioned one earlier on, but there's a couple you can choose from. Insulin is the one that I am most familiar with, but you guys can also go for human growth hormone or something similar as well. Okay, number 3, scientists use bacteria to act as host cells to produce enzymes commercially by the process of genetic engineering. Name the structure in the source cell from which the required gene is extracted during the first stage of this process. So the structure is called the chromosome. Okay, the chromosome. Next step is to describe the stages of the process that would be used to produce genetically engineered bacteria after the required gene has been extracted from the source cell. So basically they're saying they've already extracted the required gene. What comes next? Okay, so we need to remove the plasmid, or better word is extract, isn't it? Extract the plasmid from the bacterial cell. We need to then insert the required gene into the plasmid. We then need to place or insert the modified plasmid into the host bacterial cell. And the final mark is for stating that these steps involve the use of enzymes. Okay. These steps involve the use of enzymes. So that's your 4 points that you're looking for there to get the marks in this question. Okay. Because they won't allow you to do the initial steps, the locating the required gene in the source chromosome or extracting the required gene. Those are already taken. So these are the other points that we can make there. Okay. You could go on to mention that into the host bacterial cell to make a genetically modified organism, and that it multiplies to make more copies of the bacterial cell as well. Okay, folks, so I hope those questions have helped to cement your knowledge for the genetic engineering topic. And as always, folks, if you've got any comments or questions or things you want to ask about, pop them into the comments below the video, and we'll see you next time for the respiration topic. Okay, for now, that's all. Class dismissed.